The EpiNext CUT&LUNCH Assay Kit provides a rapid and efficient method for studying in vivo DNA-protein interactions. The kit integrates all the benefits of traditional chromatin immunoprecipitation and cut-and-run, while effectively addressing their limitations. With this kit, target protein-associated DNA can be enriched in just under 2 hours. The unique DNA cleavage enzymes ensure low sequence bias, enhancing specificity and minimizing background, and the enriched DNA is suitable for both gene-specific qPCR and genome-wide NGS analysis. First, prepare the antibody-binding reactions by adding your antibody, the positive control antibody, and the negative control antibody into separate PCR tubes containing working permeabilization buffer. Then, transfer 20 microliters of your suspended cells into each reaction tube. Also transfer 20 microliters of your suspended cells into an empty PCR tube and set it aside. This will serve as your input or no antibody control. Mix and rotate at room temperature for 30 minutes. After the 30-minute incubation, remove the tubes from the rotator. Add nuclear digestion enhancer and cleavage enzyme mix. Mix and rotate for an additional 10 minutes. After the 10-minute incubation, remove the tubes from the rotator. Add cleavage stop solution. Centrifuge for 10 seconds at 6,000 to 7,000 revolutions per minute to pellet the cell debris. After the spin down, remove the tubes from the centrifuge. Transfer the supernatants to new PCR tubes. Shake the affinity beads to completely resuspend them, then add the resuspended beads to each tube. Mix and rotate for an additional 20 minutes. After the 20-minute incubation, place the tubes on a magnetic stand, wash each reaction tube 3 times with wash buffer, and once with protein digestion buffer. Each buffer wash can be performed by first removing and discarding the solution from the reaction tubes, then adding buffer and gently pipetting up and down several times. Place the tubes back on the magnetic stand for 1 to 2 minutes to pellet the beads, then remove and discard the solution. Prepare protein digestion solution by diluting proteinase K with protein digestion buffer. After the final wash, remove the tubes from the magnetic stand and add the prepared protein digestion solution to each reaction tube, including the input control tube. Mix and incubate at 55 degrees Celsius for 15 minutes in a thermocycler. After the 15-minute incubation, place the tubes on the magnetic stand until the solutions are clear. Carefully transfer the solution from each tube into a new PCR tube. To each tube, add DNA purification solution, followed by 100% isopropanol. Resuspend the DNA binding beads by vortexing. Add the resuspended beads to each tube and mix thoroughly by pipetting up and down at least 10 times. Incubate for 5 minutes at room temperature to allow the DNA to bind to the beads, then place the tubes on the magnetic device until the solution is clear. Carefully remove and discard the supernatant. Add freshly prepared 90% ethanol to the tubes, then carefully remove and discard the ethanol. Repeat once for a total of 2 washes. Resuspend the beads in elution buffer and incubate at room temperature for 5 minutes to release the DNA from the beads. Capture the beads by placing the tubes on the magnetic stand until the solutions are completely clear. Transfer the solution from each tube into a new PCR tube. The enriched DNA can now be stored at -20 degrees Celsius or directly used to either construct DNA libraries for NGS analysis or perform a simple qPCR assay. With the EpiNext CUT&LUNCH Assay Kit, you can achieve fast, sensitive, and specific enrichment of target protein-associated DNA. For more information, please visit epigentech.com or contact your local distributor.